Hi, I'm going to be discussing antibody-mediated effector functions. And so antibodies that are produced by B cell activation help protect our bodies from pathogens in four main ways. Those ways are neutralization, opsonization, complement fixation, and something known as ADCC, which actually stands for antibody-dependent cell-mediated cytotoxicity. A mouthful. And so collectively, all four of these mechanisms are going to ultimately result, whether it's directly or indirectly, in the destruction of pathogen. And the ability of these antibodies to mediate these effector functions are based on that antibody's antigen specificity as well as that antibody's isotype. And what this isotype is going to determine is whether that antibody can actually fix complement and actually to which Fc receptors it will bind, as well as to the ultimate cellular functions it will cause. And so to start out with, I want to take an in-depth look at each of these processes, and I want to start with neutralization. So what's happening in neutralization is that antibodies can actually neutralize a pathogen by binding to that pathogen, and when it binds to it, it's going to block receptors that the pathogen normally uses to gain entry into the cell and to harm it. And so a more in-depth look is that most viruses and some bacteria, how they gain entry into cells and the ability to harm them are by binding specifically to proteins that are on that cell surface, and normally this binding will induce endocytosis. So to look at some viruses that we've probably heard about in every day, I want to talk about the influenza virus and the flu and the HIV virus. So this HIV virus actually expresses a protein known as GP120, and this GP120 will bind to the co-receptor CD4. While the influenza, the flu virus, expresses a protein known as hemagglutinin, and that's actually going to bind to some modified sugar proteins on epithelial cells. So there are antibodies that will bind these proteins and in this response, and the antibodies that do this are actually very effective and very potent, because what they're doing is that they are actually preventing a pathogen from ever initiating an infection, because they're blocking the entry of that pathogen before it can actually ever do any damage. And the antibodies that do this are known as neutralizing antibodies. And pathogens that have actually been disarmed by these neutralizing antibodies are typically then going to be phagocytosed by macrophages. And so neutralizing antibodies, as well as blocking viruses that we talked about, can actually also block toxins from entering cells. And so a virus that I want to talk about that we've probably heard of is called the tetanus toxoid virus, which is actually a product of the Clostridium tetani bacteria. Excuse me. And this toxin is a neurotoxin that can actually be very harmful because it can induce muscle cellular contraction that would be uncontrolled and could ultimately result in death. But what we get is a vaccine known as the tetanus vaccine. And this vaccine actually contains an inactive version of that tetanus toxin. But that inactive version is going to stimulate our B cell to produce antibodies against the tetanus. It's going to produce anti-tetanus antibodies. And these are going to be neutralizing antibodies that can bind and potentially inhibit the entry of that very harmful toxin into our nerve cells. And another toxin that we see in which these neutralizing antibodies are working is in snake venom toxins. And it's actually been seen where these neutralizing antibodies have been quite effective therapies for a few snake bites. So at a physiological level, neutralization is clearly going to be the most effective mode of protection against pathogens of any of the four methods we're going to talk about, simply because it's going to stop that pathogen before it ever gets to any infection, before it ever enters the cell at all. But the problem that we're seeing is that because microbes can proliferate so quickly, they're actually being able to generate genetic variants of themselves. And these variants are actually able to escape and evade those antibodies that would normally neutralize them. And what we're discovering is that once these microbes, once these pathogens figure out they can escape the antibody, they're going to create more of that modified version of itself that can escape the antibody. And once they start creating that modified version, it's going to proliferate so rapidly, and that infection is going to be able to spread so rapidly. And so what I want to talk about, looking back at the influenza virus and the HIV virus, how this happens. So an example is that the flu virus will create variants, modified variants of that hemagglutinin, and those won't bind antibodies. And because they won't bind antibodies, clearly they're going to have a reproductive advantage. And like I talked about earlier, they're going to rapidly start producing more and more. And those modified ones that can escape the neutralizing antibodies are going to rapidly take over infection and eventually be very, very harmful. And a further example is in HIV. It's actually known to be one of the most skilled and proficient viruses at evading these antibody responses. And that's why it's very, very harmful. And the reason that is well, in theory, that GP120 protein on the virus would actually be a very ideal target for neutralizing pathogens, or neutralizing antibodies, excuse me. But the problem is that a lot of people don't have those antibodies in their systems. Only few people actually generate them. And this is in part because that GP120 region that will bind to that CD4 co-receptor is actually concealed and hidden from the body until right before the virus will enter. And so it's kind of like a surprise attack. So that's just a look at neutralization and how some viruses and toxins we've heard about go through that neutralizing process by antibodies. And now I want to take a look at opsonization. Well, what's happening here? Opsonization actually refers to the ability of antibodies to promote or enhance the engulfment of antigens by phagocytes. Opsonizing antibodies can co-antigen and interact with Fc receptors that are on these phagocytic cells. And so we know that the phagocytic cells in our immune system kind of actually have two sets of receptors that they're capable of binding to pathogens. So the first set of receptors they have will bind them directly using that innate immune system. But the second set, the set we're talking about, can actually bind antigens found in the immune complexes with antibodies. And they're doing that via those Fc receptors we discussed. So a quick summary basically before we move on of what's happening in this opsonization process is that the binding of that pathogen antibody complex to that Fc receptor that's on the phagocyte is going to induce internalization of the complex and internal digestion of the pathogen in lysosomes, aka the goal, the eventual destruction of the pathogen. And the important thing to note in that is that whole process is utilizing those Fc receptors. And so now let's talk about complement fixation. What's going on here? Well, we know that antigen-antibody complexes can induce a complement cascade. But more specifically, when antibodies associated with that complement bind to the surface of bacteria and viruses, these antibodies can initiate a cascade of reactions that will actually result in the generation of something known as the membrane attack complex, or the MAC. And this MAC can actually perforate pathogen membranes and kill those microbes that are trying to do harm to the cell. And the capacity to stimulate this complement-mediated membrane damage is actually referred to as the ability to fix complement. And it's actually a property that's specific to specific antibody isotypes. And the two it's specific to is the IgG and the IgM antibodies. So what's really happening in this complement fixation process is that antibody-antigen complex becomes bound to the complement component. And from the time it's bound, it's either then going to be phagocytosed or lyse. If it's phagocytosed, it's going to happen via C3 receptors, and if it's lyse, it's going to be as a result of a pore formation, P-O-R-E, pore formation by the complement components C7, C8, and C9. So that's just a more in-depth look of what components are actually taking part in that complement fixation. So now let's talk about the last way that antibodies can rid pathogens, and that is known as ADCC, the antibody-dependent cell-mediated cytotoxicity. Well, what's happening here is actually giving our antibodies the ability to cooperate with the cellular branch of the immune system. And what's happening is binding and recruiting the activities of cytotoxic cells. Antibodies can actually recruit the activities of multiple cytotoxic cells, and some of those include natural killer cells or granulocytes. So unlike cytotoxic T lymphocytes, these cells that have been recruited do not have these antigen-specific T cell receptors, but they do express those Fc receptors we've already mentioned, and specifically they express Fc gamma R3. So they actually use these Fc receptors to arm themselves with antibodies that will allow them to adopt that antigen specificity that they don't have but they want. Antibodies that are bound to those natural killer cells we talked about can then direct cytotoxic activity to a specific cellular target that they have, such as an infected cell that expresses viral proteins or a tumor cell that is expressing tumor proteins on its surface. So it's going to recognize those proteins on the surface and target that cell. So this whole phenomenon of ADCC is actually under intense research right now, because they're starting to see that it could have some extreme therapeutic advantages, and it has lots of potential. So right now, actually, they're specifically using monoclonal antibodies that have been developed as cancer treatments, and they found that those are thought to act in part by taking advantage of this ADCC mechanism, and they're thinking that's happening by directing those natural killer cytotoxic cells toward those tumor cell targets that they're trying to destroy. So it's already been found that this ADCC pathway can be used in cancer treatment, so they're starting to research it more and more and hoping to find more therapeutic advantages that could arise from these antibodies that are using this mechanism. And so now we've kind of gone through a little bit of each of the 4 different ways that our antibodies can eliminate pathogens, and I have a figure I want to show you that kind of sums it all up. So here you're going to see the 4 broad categories of the antibody effector functions, and you're going to see how the antibody binding can enhance the clearance as they all end in the clearance of pathogen. And what this is our antibody up here, and the region we're concerned with, we talked about, is that Fc constant region, because that Fc region can be in interaction with those Fc receptors. So first, let's talk about the neutralization over here. And what's going to happen, as we talked about, is that antibody is going to come and bind to proteins that the pathogens are using to infect the cells, and that binding is going to keep those pathogens from ever invading by preventing that pathogen host from ever binding at all. As we talked about, that's actually the most effective form or most effective mechanism, simply because it's hitting it at its surface before the pathogen can ever enter the cell. Here's the opsonization process, as we'll see, and we can see that that Fc region of the antibody is going to interact with Fc receptors on the surface of these phagocytic cells, and that's going to induce internalization and degradation of the pathogen. And here we're going to see complement fixation, which that antibody is actually going to recruit complement proteins that can directly kill that pathogen or enhance phagocytosis. And we can see how the complement fixation will result in the generation of that MAC complex we talked about that can then perforate pathogen membranes and kill microbes. And lastly, the ADCC, where the antibody will bind to those Fc receptors on the cytotoxic cells we see, such as these natural killer cells, and we're going to see how that's going to direct activity to infected cells and tumor cells. And so that's basically a summary, and feel free to use my video.